Observing change in social behaviors of mallard ducks and humans based on varying stressful environments. We hypothesize that if a mallard duck is exposed to stressful situations or stressful events are introduced into their environment, that the mallard ducks will exhibit different behavioral responses due to the activation of the VT4R gene, which is a variant of the AVPR1A gene, and increased cortisosterone levels than one that is in a less stressful environment. The effects of human disturbances on behavior of wintering ducks by Melissa Pease and colleagues is very influential in forming our thesis and our methods, which talks about human activity creates a stressful environment for wild animals, forcing them to expend energy doing tasks to avoid humans, in turn changing the way that ducks behave.
For our methods, we took to the outdoors with pencil and notebook observation of mallard ducks on the campus of Michigan State University. We began by observing, categorizing, and measuring duck behaviors on the northwest side of Wells Hall on MSU on the banks of the Red Cedar River. We observed campus mallards at different times of the day, this being when activity on campus was high and when it was low. When activity was high on campus, behaviors of aggression increased heavily between not only male ducks, but as well as male and female ducks. Ducks expend large amounts of energy trying to avoid stressful occurrences, this being human activity. This accounts for an increase of corticosterone levels in mallard ducks, known as the stress hormone, which can cause these animals to become more aggressive due to the activation of an instinctive threat behavior towards other members of the same species. As human activity around campus decreased, behaviors of aggression decreased and returned to the same levels as measured off campus in terms of aggression towards one another.
For our off-campus research, we observed the mallard ducks at Kriegel Park in Lansing, Michigan. The gender of the mallard ducks were recorded to see if aggression increased with gender during mating season, which takes place between the months of September through November. On Michigan State's campus, humans could walk right up to the ducks, whereas at Kriegel Park, humans could only remain on the outskirts of the lake. The off-campus ducks were less social with humans and exhibited less aggressive behavior due to a decreased amount of stress and human disturbances in their environment. A playback device was used to simulate vocalizations of mallard calls, female and male grunts, courtship calls, whistles, and predatorial calls of the mallard species, including red foxes and hawks. Our team observed and recorded the different behaviors of attacking and defending, head bobbing, eating, and preening as done through the behavioral classification scale in McKinney's work, and noticed that behaviors were much less aggressive than on-campus ducks.
Studies of human activity and behavior response to stressful situations were conducted during the same months as the mallard studies, this being September through October. We observed Michigan State students on campus in both stressful situations while studying and in class, and non-stressful situations while in dining halls with their friends. Facial expressions were recorded, and this idea was inspired by Dr. Masumoto's research article that discusses classifying emotion recognition through specific facial muscle movements, these including joy, surprise, anger, and fear.
Behaviors were measured at Kriegel Park and on the campus of MSU for the mallard duck. Variability of duck behaviors on and off campus remained similar for preening and vocalization, however showed major variation for aggression and courtship behaviors, this being the ducks on campus showing higher proportions of expressing each behavior.
Preening is the act of straightening and cleaning one's feathers, which seemed to be the most frequent behavior observed within the on-campus ducks. The ducks were observed to fight over food and have no observable predators at the Red Cedar River. However, there was increased aggression toward other ducks due to the increased presence of humans and the lack of real predators other than each other. We hypothesized that aggression would be higher among the campus mallard ducks and were proven right. The campus ducks showed more aggression than off-campus ducks due to the increased human disturbance and small environment. The ducks would swim right up to a human and did not seem to feel threatened by them due to the amount of humans on Michigan State's campus and amount of people who probably supply them with food. The proportion of duck behavior compared to their sample size was measured to keep the data statistically similar.
This figure shows the average distance that mallard ducks moved away from people at Kriegel Park in East Lansing, Michigan, compared to the Red Cedar River at Michigan State University. Each column on the graph categorizes humans into either walkers, runners, or bikers, and each location is categorized by the color of the bar. It was found that ducks on campus were more accustomed to humans being in the environment, and Kriegel Park ducks were more stressed to the presence of humans in their environment.
This article by Sullivan was very influential in forming our thesis as well, giving more information about the genetics. And they said that the AVTR gene shows a role in avian stress response and is approximately 70% homology with the AVPR in humans and has a functional role in determining those behaviors. The AVPR1A gene is found on the long arm, the Q arm of the 12th chromosome at position 14.2.
Through this research, we have seen that stress mediates behavior change in both species, and we can attempt to connect the two species genetically using PCR.